welcome to all of you. I'm Indu Tiwari, Assistant Professor, Siddhartha Institute of Pharmacy, Dehradun. As Nelson Mandela said, education is the most powerful weapon which you can use to change the world. So to sharpen this weapon, I want to welcome our next honorable speaker, Dr. Mohmad Shaikh, Associate Professor in Jeffrey Cha School of Medicine and Health Sciences, Monash University, Malaysia. Now I welcome Dr. Shaikh to shower their knowledge. Welcome, sir.
Thank you, Ms. Indu. Thank you again to Dr. Sanjay specifically for kind invitation and Siddhartha Institute of Pharmacy, Dehradun, and Siddhartha Group of Institutions. So coming to my topic, which is zebrafish, and as Dr. Ms. Renu rightly pointed out, zebrafish is gaining very much popularity when it comes to drug discovery, and specifically when we talk about CNS drug and some other disease conditions where zebrafish has an upper hand compared to the other animal models. So the outline for today's research, I'll be talking a bit on the translational research. I think that very much matches to the topic today. And also some timeline on drug development, which probably some of you are completely aware of, but I'm just going to give a quick snapshot of it for the benefit of our undergraduate students. And then in general, we'll talk about animals in drug discovery, why, what, and how. Specifically, we are talking about zebrafish as the main theme of this topic as well. And then examples from my own research, which is epilepsy, and what are the parameters you actually choose which mimic the disease condition which happens in humans. Some of the merits anticonvulsant screening models of epilepsy, and some chemical as well as the genetic models of epilepsy, which we have done some research on. Then zebrafish in personalized epilepsy care, which is, I think, the next thing coming up, personalized medicine, and then the success story of zebrafish in drug discovery. So, I mean, if many of you are definitely aware what translational research is, which we are talking about something in a very simple sense. We are talking about it, and so that too in a pharmaceutical drug discovery. We say from lab or from laboratory to the clinic or from bench to bedside. So it's a bi-directional process where you integrate your basic clinical as well as sometimes it also involves population policy and public health research just to translate the research from the main small research study to the public. I think and some of these research definitely impact the policies and the policy papers in different countries. And the overall goal of the translational research is to speed up the scientific discovery into communities. That's what I said. It should benefit the patient and people and improve the quality of life of the people in general. And if you look at these three color-coded different process, which is basic research, translational research, and clinical research. In basic research, we do understand how does it work. So whenever, even undergraduate or postgraduate research we do in our university or schools or institutions, we're trying to find out how a particular drug is working or what is the mechanism it works through, what specific receptors or what specific targets is targeting. That's all about it. Then we are talking about something called I'm moving to the clinical point, which is let me find a pointer here. Okay, so we are talking about what are the effects and how well does it work, whether it's really translated into the clinics, can it be used in patients. I think Dr. Feroz has explained like the effect of this Candida fungus infections, and where drugs like atorvastatin and ciprofloxacin can be in scientific study or lab-based study found that it has some impact and some interaction, I will say, which lesser the effectivity of one or other drug, but whether it is can be really mimicked in the clinical condition, can that effect similarly seen in patient. So we are talking about from basic research to moving to the clinical research. And then in translational research, we do ask this question, whether this basic research can be translated. I think that's really, very important. Any basic research which is which do not have a translational value. So it's, I mean, you will end up in, publication, or you end up in thesis of some students, but it may not have a translational value. So the question, the legitimate question we're asking here is, in translational research, can it be applied to people? And I think, again, for the knowledge of each and every postgraduate student, I will say, whenever you do a research, you need to think whether my research is translational or not, whether I can imply the outcome of this basic science research into patients. All these questions, I think, are very important before you start a simple experiment in your lab. What I'm trying to say, you need to know the bigger picture of your own project or own experiment before doing it, so you have a very clear-cut idea of what you're trying to achieve. This is a typical drug development timeline. You might have seen several pictures, but what I'm trying to show, the drug development or drug discovery process is a lengthy process, which billion of dollars involved in that, and then it involves typical steps like the drug research. So I think most of the laboratory-based or even the research which are done in university settings are basically just lie there in the drug research and preclinical phase. And very rarely it moves to the clinical trial. But when industry works, they works with hundreds of thousands of compounds, and they go proper from the target-based to cell-based to animal-based research when they do this drug research and preclinical trial. And in preclinical trial, you're able to filter about 250 compounds, which may have some hits, and then it further translated into clinical trial. There are a lot of process of investigation, new drug and new drug applications, which are applied at different stages, and then it goes into clinical trial. I'm not going to go in detail because I think many of you are aware of the phase 1, phase 2, and phase 3, which depends on healthy volunteers and a low population of patients than the real patient, looking at different safety as well as efficacy of the drug. And finally, you get one drug which get approved or go through all these phase 3 trials and get into the market. But when they are in the market, we still call it phase 4 studies or pharmacovigilance studies, because any drug which may exhibit any serious side effects that can be withdrawn from the market. So although many drugs are in the market, but they can be withdrawn if they show a critical or serious side effects. And the timeline, if you look at it, is about 15 to 18 years in order to a drug to come into the market. But I think a lot of new technologies and advances in drug discovery research, this timeline may be shrinking, and maybe in future we may see some drugs may develop even in a lesser time. And this is, again, very important from drug discovery point of view to understand target-based versus phenotype-based drug discovery. And when I say target-based drug discovery, the specific target. For example, a lot of research has been done, the current scenario, which is COVID-19, and people know, what type of protein, what kind of receptor, what kind of shock proteins which are actually there in the coronavirus, and then knowing those specific target, then you target them. Similarly, it also applies to a lot of receptors or enzyme-based assay where target. For example, if I give example of Alzheimer's disease, acetylcholine esterase, beta amyloid, which are very well-known target, and if you target them, there is a chance that it will give you a disease-modifying effect. But sometimes, some of the cases, you only have phenotype-based drug discovery. And again, I'm giving example of epilepsy, where sometimes the objective of drug or therapy is control seizures because the root cause of the disease is still unknown. And this is where the phenotype-based drug discovery actually is also another path or another way of drug discovery. But the ultimate goal is to improve the quality of life of the individuals and the patients. And moving on, looking at the animal models in drug discovery, and I'm sure many of you are very much aware, right from using higher species, especially dogs, which was very popular, I will say, animal model for many disease condition. And then it moved further to some of the chicks, rodents, as well as primates research. And this is again based on the similarity they have from the human. We are talking about similarity with the system. We are talking about similarity to the disease condition. We also talk about the cost involved in the research, whether these animal models are really viable, whether you can do a large screening using this model. So I think based on that, from 1900 to till date, we have so many model organisms which are used in different diseases. And I'm not going to go in detail of it, but just to give a quick example, like the rodent models, or rats models specifically, are very helpful in studying hypertension or diabetes in which mimic a clinical condition to the human diseases. But I think as we are revolutionizing the animal model, from this higher model, we are moving to more cell-based and the small organism-based assays. And this is where zebrafish, fruit fly, Drosophila, or larvae or worms actually are gaining popularity. And one of the reason is their genetic similarity. So that's the reason. If you see zebrafish sitting up high there compared to Drosophila and worms, but I'll go in detail of it in a bit. And again, very, very important principle, which is 3R, which is refine, replace, and reduce in animal use in drug discovery or experimental studies. Again, very, very important. And this is where the non-mammalian small models like zebrafish, Drosophila, and others are actually getting popularity in that. So that's in a nutshell, like the whole picture of animal model in drug discovery. And again, moving to specifically zooming out on the models which are very popular these days, which are smaller non-mammalian models. And this typical figure explains the closeness to the human genetic makeup, with all of these organisms. And we see, Drosophila and Danio, which is zebrafish, are much more closer. And zebrafish obviously much more closer because it shares about 80% of genetics makeup similar to the human, and about 84% of the genes are of the disease gene actually also present in zebrafish. So that makes a zebrafish a popular model when it comes to the human disease drug discovery testing in animal models. So this is something which was published in 2010. And if you look at the mammals or rodent models or other models which were very famous in the drug discovery, were majorly mice and rats. It take about 90 to 95% of the share. And other animals like guinea pig, rabbits, hamsters, cats, dogs, ferrets, and non-human primates, they have about a lower percentage compared to the mice and rat. But having said that, the scenario is changing. And if you look at the popularity of zebrafish or other fishes specifically are getting more population, close to 30% of the share. So they are, taking away the share which rats was having, and the share of the rats is reducing around 8%. And again, mice is still very popular. Again, compared to the rats, they are much more smaller. And a lot of models are actually can be well studied in mice, which are non-genetic. But when we talk about genetic condition, I think, and also the phenotype screening, where zebrafish is actually stealing some of the share from the mice and rats. Having said that, I was talking about the similarity between humans and the zebrafish. And as I mentioned, about 70% of the normal gene and 80% of the disease-associated genes are found is found in zebrafish, which is similar to humans. And it contains all the major organs when we talk about, brain, liver, pancreas, gallbladder, circulatory system, digestive system, and the lungs or swim bladder, which are actually very much similar to the human, humans. And all the diseases which are related to it can be studied in zebrafish. So if you talk about whether cancer can be studied, can be lung cancer or pancreatic cancer or other cancer can be studied in zebrafish, the answer is yes. But I think the zebrafish getting more popularity in a lot of central nervous system disease, and that's our today's topic, and I'm going to emphasize more on. So why zebrafish? We get this question very often, and if you look at the reasons which I have put in this picture, which we have published in Frontiers in 2017, it says it's a living organism. So when a lot of disease can be studied in vitro method, but I will give an example of especially the CNS drug. If you find a drug is very effective in neuronal cells, and you feel okay, this could be helpful in animal models or in patients, but when it was tested in dynamic system like rodents or humans, that drug didn't work. And one of the reason is stringent blood-brain barrier, which is found in brain, which does not allow each and every drug to cross. And this is where if you just test in vitro system, that will not allow you the opportunity to check whether this drug has an ability to cross blood-brain barrier. Whereas if we use zebrafish, which has a similar blood-brain barrier stringent system that of human or other rodents, then that makes it a perfect model. If any drug which shows efficacy or effectiveness in zebrafish model, it is very or highly likely that drug actually can be effective in rodents as well as in human models as well. And that's why living organism is again very, very important. It's a diurnal species. That means zebrafish has a similar sleep cycle as that of human. It stays awake in the day and sleep at night, whereas rodents, rats and mice, it is an nocturnal animals, and they sleep in the daytime and they are active in the nighttime. So that's, I think, again, one of the important point which makes zebrafish as an model of choice. You can do PK/PD studies, pharmacokinetics and pharmacodynamics. When I say pharmacokinetic, actually researchers have developed very advanced technologies, even with very small amount of blood which you get from zebrafish, about 10 microliter or something, but you will be still able to study some of the pharmacokinetic parameters from that. Blood-brain barrier permeability, yes, because it does have a very stringent blood-brain barrier, and it allows us to study maybe permeability. You can do gene expression studies, mRNA expression studies. It is cost-effective, so very cheap. You can house. So if you compare from rats to mice, I think the ratio is 1:10 or 1:20 in terms of cost. So it's very cheap compared to rats and mice. And when it comes to high throughput screening, you need, as again, a very low volume of drug, similar to the cell culture models. So that's actually make it more popular. The last point is it is definitely rapid compared to the rodents models, and it is very reliable because it is a whole living organism system. So I hope that gives you an idea. But when we talk about why zebrafish, that again, it says it conserves most of the targets of the human diseases. It conserves drug metabolism. That means in the liver there are enzymes. So a disease related to that can be also studied in zebrafish. It conserves physiology. You talk about neurophysiology, you talk about sleep cycle. All those physiologies are well conserved in the zebrafish as well. And when you talk about conserved pharmacology, that means different diseases and the effect of the drugs are actually well studied and can be compared with the human data in zebrafish. So overall, that makes zebrafish again a very interesting and promising model. This is typically the cycle of the zebrafish development. Within 90 hours or 96 hours, actually zebrafish are fully grown. So those diseases which are like developmental diseases or neurodevelopmental diseases can be easily studied in zebrafish model. So if for example, if you want to study a drug which has ability to defect the heart growth in fetus, you can easily study, because you can completely capture the development of heart, the completely development of zebrafish using the video cam mounted on the this petri dish where you're keeping this, and then whole life cycle of the zebrafish can be studied, especially the growth life cycle. And this is where it gained popularity as a model organism to study neurodevelopmental or developmental diseases. So yeah, and then this is also given opportunity where you can do genetic mutation in the embryo, and then when you get zebrafish, you probably find it knocked in or knocked out. Definitely, there are a lot of other further work required, but in general, the success rate of the genetic mutation is quite high in zebrafish models. So I was talking about the similarity in the genetic. I think I'm not going to spend much time, but I think this figure completely explain zebrafish and humans share about 70% of the protein coding human genes in both of these organisms. So next question is why zebrafish? I think again answering that, because you can create the CRISPR mutant using zebrafish, which are much more easier compared to the other rodent models. Again, which saves time, money, and the duration, which is similar to time, but also the efficacy of the assay as well. Because zebrafish, you get about 200 to 300 embryos in one shot, compared to rodents where you get about maximum 10 to 12 pups only. So that's again very important, I say, merits of zebrafish to use it for the drug discovery and high throughput screening in particular. So you can do molecular assay, you can do cellular assay, you can also do behavior analysis, and I will share further in the examples which I'll be showing in the next few slides about the behavior analysis using the zebrafish model. So one of the important aspect of CNS drug discovery is the behavior analysis. And if you ask me, in rodents, we do learning and memory tests, for example, we do seizure analysis, we do depression behavior, we do aggressive behavior or anxiety behavior, and a lot of other studies, Alzheimer's disease or Parkinson's disease, which are models in rodent models. Can this be a model in zebrafish model? The answer is yes. And there are a number of models which are published in very high impact journals where zebrafish actually successfully can be studied to different behavioral aspects of the disease. For example, if you talk about Alzheimer's disease, you can check learning and memory using zebrafish in T-maze, which is similar to the plus maze in rodents, those who have used, or passive avoidance test, which is again similar to the rodent models, and the social preference and other models, so on and so forth. So the good news is, yes, you can use the models using zebrafish. You can do the behavior analysis using automated system, and I'll come to that in a minute. So if you look at the timeline, how much zebrafish has contributed to the CNS drug discovery, and you can see right from into the early behavior of learning and memory to the drug addiction studies, cognitive aging, epilepsy, anxiety, reward pathway analysis, and the psychosis, hallucinogens, and drug withdrawal testing, Alzheimer's disease, Parkinson's disease, antidepressant, selective serotonin reuptake inhibitors for the pain, for autism, depression, antipsychotics, cognitive enhancer, so on and so forth. All those actually drug discovery tools were actually kind of established in zebrafish model.